All right, hello everyone. We will be covering chapter 22 today, which is the reproductive system. It's our last chapter in AMP2. All right, so here are some roots, prefixes, and suffixes that you will need to learn for this chapter. I'll read those to you. Some of them you'll already know, some of them you won't. All right, so let's get right into it. So first is meiosis. So first, we have these things called gonads. Both males and females have gonads. They are just essentially organs that produce hormones and gametes. Gametes are just sex cells, and males' gonads are called testes, and they produce sperm. And in females, ovaries are the gonads, and they produce egg cells or ova. Ova is plural. Ovum, O-V-U-M, is singular. And we also call them oocytes. You actually pronounce both of the O's, oocytes, or oocyte would be singular. And the sex cells have one set of genetic instructions on 23 chromosomes. Remember, humans have 23 chromosomes. But in regular body cells, we have two sets of genetic information on 46 chromosomes. And so, the sperm has 23 chromosomes and the egg has 23 chromosomes. And when the sperm and egg unite, that's how we get the 46 chromosomes. 23 times 2 is 46. All right. The female, the egg, always has an X chromosome, and the male either has an X or a Y chromosome. Half the sperm have an X chromosome, and half the sperm have a Y chromosome. So you can see by the little Punnett square up in the top right-hand corner that basically there's a 50-50 chance that the child will be female and a 50-50 chance that the child will be male. So it's actually the male that is responsible, for lack of a better word. The male is who determines the sex of the child. And when we talk about replication of the sex cells, that's called meiosis. If you remember from AMP1, we talked about mitosis, where we had, the interphase, prophase, metaphase, anaphase, and telophase, or telophase. That was with regular body cells, non-sex cells. But when we talk about sex cells, we are talking about meiosis, not mitosis. So in meiosis, we actually can break that down into meiosis 1 and meiosis 2. It's actually two consecutive divisions. So we still have, the chromosomes are going to replicate, and they still have, the chromosomes are going to be connected at the centromere. So remember, if we're talking about, like the X, where the X crosses, that's going to cross at that centromere. And if you remember, the spindle fibers that come out of the centrioles are going to attach at the centromere. And we do this because otherwise that fertilized ovum will have too much genetic material, which we'll talk about a little bit later. Now we use this term called diploid and haploid. So diploid just means that it contains two sets of chromosomes in 23 pairs. Haploid, which means that it would just contain one set of chromosomes. So when we have the egg and the sperm that unite, each cell, each egg and each sperm would be a haploid, and when they come together, they would have that would make a resulting cell of a diploid cell, so that you have half your genetic material from one parent, half of the genetic material from another parent. And if you have one set of 23 chromosomes and one set of 23 chromosomes, when they come together, now you're going to have two sets of chromosomes in 23 pairs. But when we do the whole replication process, we have to replicate first these chromosomes that are in either the egg or the sperm. And so now we're creating a diploid cell that contains two sets of chromosomes that have 23 pairs. And we'll see why that is in just a second. So in meiosis 1, we're actually going to kind of separate out those two sets of pairs into one set of pairs. So that meiosis 1, it says separates homologous pairs of chromosomes. And so these homologous just means the same, or gene for gene. So they're not necessarily going to be identical, only because those genes may have variants. And we'll see in a couple slides what those variants are. But when we do separate out those chromosomes, because remember, we're in a diploid, we have a diploid cell because we've replicated, and we have, two sets of chromosomes. And after meiosis 1, we're going to take two sets of chromosomes and separate that cell into one set of chromosomes. So we're going to take a diploid cell and make a haploid cell. So after meiosis 1, we're going to take two sets of chromosomes of 23 pairs, and we're going to turn it into one set of chromosomes of 23 pairs. So, and then in meiosis 2, we're going to take those, that one set of chromosome of 23 pairs, and we're going to separate that one set of chromosomes, and we're going to break it up into those chromatids. So remember that X that we saw in AMP1, we're going to take that X in meiosis 2, and we're going to break up that X into the one strand of the X and the other strand of the X. All right, so homologous chromosomes. Here you can just see just a picture of the homologous chromosomes. So you can see that, get my little pen out. So here you can see the pairs of homologous chromosomes here. And in right before meiosis 1, we've actually replicated those pairs of homologous chromosomes. So this is prior to any type of cell division or movement of chromosomes, or the spindle fibers or anything like that. So the first step is to take your genetic material and to replicate that genetic material. So in meiosis 1, we're still going to follow the same pathway that we did for mitosis, prophase, metaphase, anaphase, telophase. And you do need to know these stages in order. So when in meiosis 1, what we see during prophase is that the homologous chromosomes pair up. And that is, it says, involves synapsis. So these pairs of chromosomes are going to kind of kind of go together. You know, chromosome 1 is going to go together. Chromosome 2 is going to kind of pair up together. Chromosome 3 all the way to 23. They're gonna pair up. And what's going to happen is these chromosomes are going to kind of like cross over. They're going to kind of overlap or interweave with each other. And I'll just kind of show you on the next slide. So you can see here that these kind of chromosomes are going to overlap. And you can see that, you know, the result is that some of the green goes over into the blue chromosome and some of the blue goes over into the green chromosome. And the reason why we have this, it's called crossover and recombination. And the reason why we have this is because if we did not have this, those new cells, you know, once we do the whole meiosis thing, if we did not have crossover, the new cells would not be genetically variable. There would be less genetic variability because some of the chromosomes would be entirely maternal and some chromosomes would be entirely paternal. And I'm sure, you know, some people would agree that, you know, they don't want their kid to be exactly like their dad, or some, you know, people might agree that they don't want their kid to be exactly like the mom, in how they look, stuff like that. So that's why we have this crossover, so that so we can have some genetic variability so that the offspring are not exactly like the parents. All right, so, and then we still kind of follow the same path as we did for mitosis. In metaphase 1, those chromosomes are going to line up. In anaphase 1, the chromosomes are going to start to separate. Remember, the spindle fibers come down, attach to the centromeres, and then the chromosomes themselves are going to separate and go to the opposite sides of the cell, and this is going to reduce the chromosome number by half. And then in telophase, or telophase, however you want to pronounce it, that's when the cell actually kind of divides from one cell into two. So we're still going to have 23 chromosomes and 46 chromatids. And we'll see a picture of this later on. So in prophase 1, all right, so in prophase 1, remember we've done our DNA replication. So we actually have 46 chromosomes and 92 chromatids. So remember, if we have an X, that's one chromosome, and each strand of the X is a chromatid. So if we have one X, we have two chromatids. So does that make sense? So in prophase 1, we have 46 and 92. You know, the same with anaphase and metaphase, or I guess metaphase and anaphase, 46, 92, 46, and 92. But at the end of meiosis 1, once we split those cells, we're going to, because we have split those cells, we've split the number of chromosomes. So now we're left with 23 chromosomes and 46 chromatids. So overall, the overall result of meiosis 1 is basically that the homologous chromosome pairs separate. So here's just another picture example of one. You can see first that we have duplicated the number of chromosomes. And 2, you can see the crossover that has occurred. So you can see that some portions of the chromosome have the yellow and some portions have, you can see the genetic variability because the chromosome isn't all blue and this chromosome isn't all green. So you can see the crossover that has occurred. And then we have done meiosis 1. So this is meiosis 1 here. And then you can see that essentially all we've done is broken this cell in half. So, obviously this is just one cell, or that it's a cell that has one chromosome. But in humans, remember, we have 23 chromosomes with 46 chromatids. So, that would be the result of meiosis 1. And then I know we haven't talked about meiosis 2 yet, but here's where we would do meiosis 2. And in meiosis 2, essentially what we would be doing is splitting the cell. Is just splitting the cell down here, so that at the end of meiosis 2, we would have 23 chromosomes with 23 chromatids in each cell. So each cell would have 23 chromosomes and 23 chromatids. All right, so we'll talk a little bit about meiosis 2 now. So meiosis 2 begins right after the end of telophase 1. And meiosis 2 just follows exactly the same thing as what happens during mitosis. We have prophase, and metaphase, anaphase, and telophase. Where in prophase, just like in mitosis, where the chromosomes condense. In metaphase, the spindle fibers are going to come down. The chromosomes are lined up at that equatorial plate. In anaphase, the centromeres separate, and now those chromosomes separate, and those chromatids, because those chromosomes are separated, those chromosomes are no longer called chromosomes. We call them chromatids. So the chromatids are going to move to the opposite sides of the cell. And then in telophase is where we have that cell division, and telophase would split off that cell. And, like I said, we have now 23 chromosomes and 23 chromatids. Now, one interesting thing is if a spindle fiber fails to attach to a chromosome at the centromere, one of the daughter cells, remember a daughter cell is, one of these cells, one of the daughter cells would contain extra chromosomes, and one of the daughter cells would contain less chromosomes. So here, let me just get rid of some of my pen markings. So let's say that in this cell, we have our spindle fibers, and let's say the spindle fiber doesn't attach to this, doesn't attach to the centromere right here. This chromatid would actually be over here in this cell. So this cell would actually have an additional chromosome, and this cell would have one less chromosome. And we'll get into different scenarios of what would happen what would happen as a result of this as we progress through the lecture. All right, so as we saw, in this slide up here, we start out with one cell prior to meiosis 1. And then at the end of meiosis 2, we produced 4 gametes. And if we look at all of the different possible chromosome combinations in gamete, there's actually 8 million possible chromosome combinations in a gamete, which is why we have so much genetic variability in the world. And like I said, so this occurs in sperm, but not in eggs. And we'll talk about this a little bit later on. In eggs, we don't have this one cell that produces 4 different egg cells. In sperm, we have this one cell that produces 4 different sperm cells. But in eggs, the process is a little bit different. And we'll talk about, how the egg kind of goes through this process a little bit later on. But what we find is that, males produce more gametes than females. Males produce more sperm than females produce eggs. And we'll see the numbers, towards the end of this lecture. But, males over their lifetime are going to produce millions and millions of sperm, whereas females are only going to produce a few hundred eggs over the course of their lifetime. And male gametes are smaller in size when compared to female gametes. Well, sperm are much smaller in size than eggs are. So, and as we've said before, this division ends with sex cells having one set of genetic instruction. We're going to have 23 chromosomes compared to the 2 sets in other cells. And like I said, the reason why we have that is because we have 23 chromosomes in the sperm, 23 chromosomes in the egg, so that when sperm and egg unite, that's when we're going to get the 2 sets or 46 chromosomes for the offspring. So in the previous slide, I mentioned that, there's this little statement here about, what happens when the centromere or when the spindle fiber fails to attach at the centromere. We're going to have either extra chromosomes or not enough chromosomes. So what do you think? So let's go back also to so one thing that I didn't say is when we're talking about the different divisions, when we're talking about the first mitotic division, that's also called primary mitotic division, and secondary mitotic division is called secondary mitotic division. And the result, if we're talking about sperm, I could say primary spermatocytes. And if I'm talking about primary spermatocytes, I'm talking about sperm cells that are produced after the first mitotic division. So if I'm talking about primary spermatocytes, I would be talking about sperm cells that have 23 chromosomes and 46 chromatids. But if I'm talking about secondary spermatocytes, I would be talking about sperm cells that would fall after telophase 2 that have 23 chromosomes and 23 chromatids. All right, so we have to know the difference between, primary and secondary. All right, so now that we have that out of the way, what do you think will happen if primary spermatocytes do not divide into secondary spermatocytes, but they still undergo spermiogenesis and form mature sperm? How will the resulting sperm differ from normal sperm? So we have sperm cells. So it says we have primary spermatocytes that do not divide into secondary spermatocytes. So let's go back to this slide. We'll do some erasing. Oh, that's not erasing. Okay. All right. So, and I guess I could put my numbers back up here. So at the beginning of our first meiotic or meiosis 1, we have 46 chromosomes and 92 chromatids. And remember, we are a diploid, and here we are haploid. So haploid would be 23 and 46. All right, so the question that I asked was, what do you think would happen if primary spermatocytes do not divide into secondary spermatocytes, yet they still undergo spermiogenesis and form mature sperm? How will the resulting sperm differ from normal sperm? So if we look at this slide, here's where we had meiosis 1, and here is where we had meiosis 2. So the question says, we were successful in this stage, but we stopped here. So remember, if we go back to our numbers, at the end of meiosis, we had 23, and after the end of meiosis 1, we had 23 and 46. So at the end of meiosis 1, the sperm cells should be haploid. If the primary spermatocytes do not divide into secondary spermatocytes, they are going to be diploid instead of haploid. Remember, I said that the question that I asked was, what do you think would happen if primary spermatocytes do not divide into secondary spermatocytes? So here, this is kind of where we stopped. That cell did not divide. So we kind of stopped after anaphase and we didn't do telekinesis 1. So that resulting cell is going to remain diploid with 46 and 92. Now, let's assume that the sperm does fertilize an egg. what's going to be the chromosome count of that zygote? Remember, a zygote is like a fertilized egg. So sperm plus egg equals zygote. So what do you think the sperm count, or what do you think the chromosome count would be of that zygote? Well, if we go back to what should happen at the end of telophase 2, or at the end of telophase 2, remember we have 23 chromosomes and 23 chromatids. But here we have 46. So basically 46 from the sperm, 23 chromosomes from the egg, so we would have 69 chromosomes. So the egg will be haploid. The egg is going to be haploid because the question didn't say, the egg was messed up. So the egg is going to be haploid. It's going to have 23 chromosomes. The sperm is going to be diploid because remember, it did not successfully divide and it didn't successfully do telekinesis 1. So the sperm has 46 chromosomes. The egg has 23 chromosomes. The zygote will now have 69 chromosomes instead of the normal 46 chromosomes. All right, this is just a picture of what happens. Remember, interphase is when we replicate the DNA. So here we are actually replicating the whole chromosome. And then in meiosis, we have prophase. And of course, we didn't talk about prophase 1, metaphase, anaphase, telophase, and then in meiosis 2, prophase, metaphase, anaphase, telophase, so that, we start out with one cell and we end up with 4 daughter cells that are identical genetic information to, or not necessarily homologous, not identical, to the originating cell. Here's just another picture. It just shows, another picture of meiosis in humans. All right, so moving on, male reproductive organs. I think most of you know by now what the male reproductive organs are. Maybe not all of them, but we know the gist of what the male reproductive organs are. We'll go through them a little bit more in depth. So the male, remember, the gonads in males are the testes. Those are the primary sex organs. The testes are. And the testes produce those sperm cells called spermatozoa. And those are the testes produce the male sex hormones. We also have secondary sex organs or accessory sex organs. And they can be internal or they can be external. They can secrete hormones and they transport sperm and secretions to the outside of the body. So here you can see just some of the major ones. The ductus deferens or the vas deferens. That's actually what they snip if you're having a vasectomy. Here is your testis. or testes would be plural. The penis, urethra. The urethra actually serves a dual purpose. Not only does it allow you to pee, or it conveys urine from your bladder to the outside world, but it also provides for sperm movement upon ejaculation. So here you can see your ejaculatory duct. So from the testes, the sperm would move up the vas deferens, and here through your the ejaculatory duct into the urethra. And we'll talk about the different sex organs. But first, with the structure of the testes are about 5 centimeters long, 3 centimeters in diameter, and they're surrounded by this thing called a tunica albuginea or albuginea. Most people say albuginea. And it's just like a tough fibrous capsule that encloses each of the testes or each of the testes. And the testes can actually be kind of divided into lobes. And there's about 250 lobes in each of the testis. They're just compartments separated by connective tissue. And within each of the 250 lobes, we have these things called seminiferous tubules. So you can see up here, the different lobes. Here you can see the seminiferous tubules. And these seminiferous tubules, they're basically just a bunch of coiled tubes inside the 250 lobes or lobules. And they're lined with a specialized stratified epithelium that contain these spermatogenic cells. So spermatogenic just means sperm creation cells, and these things called sustenticular cells or Sertoli cells that, give rise to sperm cells, spermatogenic cells. And damage to the sustenticular cells will result in starvation of spermatogenic cells. They will cease development because they won't receive the nutrients or support that they need. All right. Testes also have these things called interstitial cells or cells of Leydig. You can see those up here. And these cells of Leydig or interstitial cells, they just produce your androgens, the male sex hormones, the androgens. The ret testes, they're the channel that transports the sperm from the testes to the epididymis. Let's see if it's back on. So here you can see the epididymis. So it would just the ret testis would transport the sperm from the testes to the epididymis. And then the epididymis is another coiled tube that transports the sperm from the ret testes to the vas deferens. So you can kind of see that here. The epididymis transports the sperm to the vas deferens. All right, so descent of the testes. So in the womb, the testes aren't descending in like the testicular sac like we see them, when you're an adult. They're actually formed up inside your abdomen. And then about 1 to 2 months before birth, we're going to have some release of testosterone that's going to guide the testes down through the abdomen into the scrotal sac. And there's this thing called a gubernaculum, or it's a fibrous cord that helps guide this sac through the inguinal canal. Um, and then after descent, the spermatic cord's going to develop along with the vas deferens, blood vessels, nerves, which eventually is going to form parts of that spermatic cord in the scrotum. But as the testes descend through the lower portion of the abdominal wall, or the pelvic wall, it can create this pouch or opening in about 25% of males, where intestines can actually kind of enter into, producing, it's called an indirect inguinal hernia. And if the intestines become constricted, blood supply can stop and it becomes what's known as a strangulated hernia. And without, prompt treatment, that strangulated hernia or those intestines, that intestinal tissue might die. Now, if the testes, it says the testes usually descend 1 to 2 months before birth. Now, if the testes don't descend or have a delayed in its descent, it can actually cause infertility. Because the testes are now exposed to a higher internal body temperature, which will slow or stop sperm development. It's actually called cryptochytism. Cryptochytism. You don't need to know that. And which is also why, for males, this, when you go into cold water, your testes actually kind of go up, closer to your body to kind of preserve heat or to get more heat. And, when it's very hot outside, your testes actually kind of get away from, or when your body's really hot, your testes kind of go away from your body. Because your body, your testes, they don't want to be near hot places because it can cause infertility. All right, some of the male internal accessory organs, they're going to nurture, transport sperm cells, which we've already talked about. They're the epididymis, ductus deferens, ejaculatory duct, seminal vesicles, prostate gland. We didn't talk about the bulbourethral gland or the urethra. We did a little bit. Epididymis, the plural is epididymes. I guess first I should say with the with here, consists of the epididymis. You're going to have to know which ones are bilateral or paired and which ones are unpaired. So in the male, we only have, actually I'll do my little eraser. So in the male, we only have one urethra. We only have one ejaculatory duct. We only have one prostate gland, and the rest, we have two epididymes, we have two vas deferens, we have two seminal vesicles, we have two bulbourethral glands. So you're gonna have to know which ones are paired or bilateral, which ones are which ones we only have one of. Epididymis, epididymes. Remember, that's that narrow coiled tube that sits on top of the testes, right here, sits on top of the testes. And it connects the duct in the testes. It runs between the testes and the vas deferens, or that rect testes and the vas deferens, and it's going to promote the maturation of sperm cells. It's lined with a pseudostratified columnar epithelium. Remember, pseudostratified columnar epithelium usually has microvilli on it, which is, in this case, non-motile stereocilia, that helps transports sperm cells into the vas deferens. All right, the vas deferens or the ductus deferens, the plural is deferentia. Um, so it's a muscular tube. It's part of the spermatic cord, and it extends from the epididymis into the ejaculatory duct. So this vas deferens, you can see, runs all the way down or all the way up, whatever you want to call it, all the way from the epididymis into the ejaculatory duct. It also has that pseudostratified columnar epithelium, and you can see that there's some cilia, little microvilli, kind of on the surface there. Seminal vesicles, or they're also glands, and they're attached to the ductus deferens or vas deferens near the base of the bladder, and they secrete a basic or alkaline fluid. Remember, it's above pH 7.0, so pH of about 8.5. Actually, I lied. pH of 7.2 to 8.0 is the pH of your seminal fluid. Has a lot of sugar, fructose, prostaglandins, and it's going to contribute most of the volume of semen. The actual volume of sperm itself is relatively minor compared to the ejaculate that does come out. Your prostate gland, it's about the size of a walnut or chestnut, and it wraps around that proximal portion of the urethra, kind of right outside the urinary bladder. Here you can see the prostate gland. You can see the prostate gland here. It actually it's like three-dimensional, so it actually wraps around the proximal portion of the urethra, just below or inferior to the bladder. Has a lot of connective tissue with some smooth muscle in it. And it secretes another, milky alkaline fluid, and it also enhances sperm motility, but it does contribute to the volume of semen. So, here you can see the prostate gland. You can tell that it is an exocrine gland only because it has a lot of those apical free surfaces. It empties its contents into the lumen. So here would be the lumen. It's going to dump its contents into these into the white spaces, the lumens. So prostate cancer, men do suffer from prostate cancer. You can typically get your prostate checked by having a medical practitioner, finger up the butt. But they do a blood test called a PSA, prostate specific antigen, is a blood test. And if you do have to have your prostate removed, prior to 25 years ago, it was this long drawn out procedure that it was a very gory procedure where they had to open you up, because it's really the prostate is not easy to get to. You have to cut through a massive amount of tissue to get there, to get to where the prostate is. But nowadays we have the robotic surgery, the Da Vinci surgery, where they kind of stick some cameras in with a little with a scalpel, and the surgeon actually sits way far over here and maneuvers the Da Vinci robot, and the robot actually performs the surgery way over here on the patient. And I actually used to love doing these surgeries. The patient is actually probably at an incline of about like that. where, the head is here and the feet are up here. And you kind of have to like Velcro strap them down, because obviously if you're at an incline about like that, you're going to fall off the table. So you really have to tape them down and Velcro strap them down so that they don't fall off. And if you have hypertension, it's something to watch out for, because, if you're putting your patient on your head, you have to can cause blood rushing to the head, hypertensive crisis, and stuff like that. So sometimes this type of surgery, the patient can't handle this type of surgery. So just monitor the blood pressure, and everything is usually okay. But because you're on your head for such a long period of time, a few hours, fluid will kind of rush to your face and will leak out. So you're going to have some edema in your face. And so I always tell, my patients' family members post-op that, just so you know, he's going to look very, very swollen in his face, and it's normal. All right. So bulbourethral glands, they're also called Cowper's glands. They lie below the prostate gland. Right here, the bulbourethral glands. They're also going to secrete some more fluid, but this fluid is actually formed in response to sexual stimulation. And it's basically for lubrication, and kind of helps everything get moving for ejaculation. But all right, so semen itself, semen is contains sperm and all of the secretions that we've talked about so far. So it contains sperm cells, it contains sperm from the testes, and then secretions from, the seminal vesicles, the prostate gland, the bulbourethral glands. It's alkaline. It's a little bit alkaline. I said, 7.2 to 8.0 here. It says 7.5. Fructose, prostaglandins, nutrients. And per ejaculate, it's about 2 milliliters to 5 milliliters. And I said, earlier on in the lecture that, males produce more sperm than females produce eggs. So with each ejaculate, males are going to ejaculate about 120 million sperm cells. And that's just per ejaculate. That's not over a lifetime. So if you think of how many times a male ejaculates over a lifetime, that's 120 times however many times they ejaculate over the course of a lifetime. And then, because of the pH of the fluid of the secretions, that's going to cause and of course the pH of the vaginal canal, that's going to cause the sperm to swim. And we'll talk about the makeup or the anatomy of the sperm a little bit later on. But the sperm has this little cap over the head of the sperm. But the sperm's going to kind of swim through the vaginal canal until it gets to the egg. And there's a simple statement here that says, sperm cannot fertilize the egg until it goes through capacitation in the female tract, which weakens the acrosome. So the acrosome is this little cap that's over the sperm, or the head of the sperm. Male infertility. Basically, there's several definitions of male infertility. Inability of sperm cells to fertilize an oocyte. It could be because the testes never descended. It could be the testes descended too late, and the temperature changes the temperature caused the sperm to be non-functional or non-motile. It could be You had mumps as a kid, and you had the mumps caused the sperm to be non-motile or non-functional. You could have poor quality sperm. You could have the abnormal acrosome on the head of the sperm. You could have poor poor flagella on the tail of the sperm. You could have a low sperm count. Remember I said, 120 milliliter or 120 million sperm per ejaculate. Here it says a low sperm count is 20 million sperm per milliliter of ejaculate. And it says per milliliter of ejaculate. Now, we learned in the last slide that, males ejaculate 2 to 5 times, or 2 to 5 milliliters per ejaculate. So here, 20 million sperm times 5 would be 100 million sperm. So, that to me still seems like a lot of sperm. So, you just kind of have to put it in context. But we do have this thing, computer-aided sperm analysis that will kind of help us more evaluate the cause of man's infertility. Is it really sperm count or sperm motility? Is it really the size or the shape of the different parts of the sperm, or is it the anatomy of the sperm? What is the cause, the main cause of sperm, of male infertility? All right, external reproductive organs, the scrotum and the penis. So the scrotum, we all know it's that pouch of subcutaneous tissue that's located behind the penis and between the penis and the anus. It does not have any adipose tissue. It lacks fat. There is this muscle called the dartos muscle. That responds to temperature changes. It's made up of smooth muscle. It's not anything that we have any control over. Remember, smooth muscle is involuntary. And this dartos muscle is what's responsible for your scrotal sac to contract or expand in response to temperature changes. I use the analogy of, going into cold water and your scrotal sac constricting, or if your body's too hot, your scrotal sac is going to relax. And we do have 2 chambers. We have a right chamber and a left chamber for your scrotal sac, and that medial septal is what divides that chamber into two separate chambers, and each chamber has a serous membrane that houses each individual testis and the epididymis. The penis itself, we all know what that is. It conveys urine through the urethra to outside of the body, but it also conveys semen through the urethra to the outside of the body. So not only does it have a urinary function, it has a reproductive function as well. And it's specialized to become erect during sexual intercourse. And when we look at the penis, we can divide the anatomy into we can divide the shaft into 3 columns of erectile tissue. We have 2 columns of this corpora cavernosa and 1 column of this corpus spongiosum. That spongiosum kind of surrounds the urethra. And we'll look at that on the next slide. We also have this thing called the glans penis. It's the distal enlargement of the corpus spongiosum. It's also known as the head of the penis. And then we also have this thing called the prepuce or the prepuce, or prepuce, however you want to pronounce it. That's the foreskin. That's what's removed if you've had a circumcision. So here you can see the 3 layers. Excuse me. The corpora cavernosa, and the corpus spongiosum. You can see that tunical, albuginea. There's your urethra, your prepuce. That's the foreskin that gets removed. The glans penis, or the head of the penis. Your external urethral orifice. That's where your pee and ejaculate comes out of. All right, so when it comes to erection, orgasm, ejaculation, so the erection is, the what you see during sexual stimulation. Remember, I've always said that, with each organ system, you have, the parasympathetic or the sympathetic nervous system takes control. Think about what happens in the middle of the woods when you meet the bear. Your reproductive system, if you're meeting the bear in the middle of the woods, getting an erection, which is your sympathetic nervous system, your body doesn't care about getting an erection while you're in the middle of the woods trying to fight for your life. So in this case, the parasympathetic nervous system is going to be the part of the nervous system that takes control. So when you are sexually aroused, your parasympathetic nervous system is going to be the primary nervous system. And if you remember, we talked about nitric oxide several times before, and nitric oxide is a vasodilator, and it's going to dilate those small arteries in the penis and going to allow for an increased blood flow, causing the erection. You're going to see the engorgement of the erectile tissue as it fills with blood, and the penis is going to swell and elongate. I will say, people with erectile dysfunction, it's usually due to an underlying disease like diabetes. There's a lot of myths out there that, too much sex will cause you to have erectile dysfunction. That's not true. Too much exercise will cause you to have erectile dysfunction. That's not true. Too little sex will cause you to have erectile dysfunction. That's not true. Erectile dysfunction is usually caused by an underlying disease, not because of, exercise or too little, too much sex. Orgasm is just the culmination of sexual stimulation. It's a pleasurable feeling of psychological and physiological release. There's it's accompanied by emission, ejaculation. Emission is is the movement of semen into the urethra, and ejaculation is the movement of semen outside of the urethra. Now, and it says dependent on sympathetic nerve impulses. Now, what's interesting is that during sexual activity, the parasympathetic nervous system is in full control right up until the point of orgasm. And then the sympathetic nervous system will take over. So, the parasympathetic nervous system will be in control until emission and ejaculation, and that's going to be dependent on the sympathetic nervous system. So you're going to have erection, emission, then ejaculation. All right, and this is just a summary slide of what we just talked about. These are some of the functions of the different male reproductive organs. All right, spermatogenesis. Remember, spermatogenesis is creation of sperm cells. Genesis means to create, and spermato is sperm. All right, so we already talked about meiosis, um, and we also have this process called spermiogenesis, where we have 4 sperm cells that are produced from meiosis, and from one spermatocyte. But spermiogenesis is how we create those spermatids, or those immature sperm cells, into sperm. And that happens in the sustentacular cells, or those Sertoli cells. And those are, just remember, large cells that are in the seminiferous tubules that support and nourish the spermatogenic cells through the development into the sperm. And remember that meiosis takes those chromosomes, so that each sperm has 23 chromosomes and 23 chromatids. So how we create sperm cells kind of follows a specific sequence. So we have this thing called a primary spermatocyte. And that primary spermatocyte, remember, would be the end of meiosis 1. Primary spermatocyte is the end of meiosis 1. And at the end of meiosis 1, there we go. At the end of meiosis 1, we would have the I'm having a little brain fart here. We would have the 23 chromosomes with 46 chromatids. All right, then then we would have so we'd have the primary spermatocyte with 46 chromosomes, each with 2 chromatids, and they undergo this meiosis 1. And during cell division, the primary spermatocyte consists of those paired homologous chromosomes. And as a result of that first meiotic division, we have 2 secondary spermatocytes that are formed. And remember, each of those would have 23 chromatids, they would have 23 chromosomes, each with 2 chromatids. Then during the second meiotic division, 4 spermatids are formed, each with 23 chromosomes, and each chromatid now represents an independent chromosome. And these spermatids that are produced are now the sperm cells. So we have the spermatogonia to the primary spermatocytes, which are diploid. Then the secondary spermatocytes, which are haploid. Then the spermatids, then the spermatozoa. And all spermatogonia are just undifferentiated spermatogenic cells, or male germ cells. All right, so because they are undifferentiated spermatogonia, they can replenish themselves, and they can give rise to spermatocytes, which develop into sperm. And then as the stages of spermatogenesis continue, the sperm migrate from the outer edge of the seminiferous tubules into the lumen. And those sustentacular cells, remember I said, they're going to provide nourishment to the developing sperm. So here's just also just a picture representation of of the mitosis, not meiosis, but the mitosis in spermatogonia. And here, for the structure of a sperm cell, it kind of looks like a little tadpole. You can see the head on the sperm. This head is also called an acrosome. It's mostly a nucleus. It does have the 23 chromosomes, and that head or the acrosome is just like a cap over the nucleus. It has a bunch of enzymes that are able to penetrate the layer of the oocyte during fertilization. And prior to fertilization, that head is going to have to be disintegrated so that the chromosomes can unite with the egg. There's also a mid piece of the sperm that contains a lot of mitochondria. Remember, mitochondria, powerhouse of a cell. It's going to produce a ton of ATP. Basically, the sperm is going to use a lot of energy to swim up the vaginal canal into the fallopian tubes. Usually, the egg is fertilized in the fallopian tubes. So the sperm has a lot of swimming to do to reach the egg. And then the third part is the tail. The tail is a flagella, is plural, flagellum is singular. Has a lot of microtubules in there that will propel the sperm forward. Here's just a microscopic view of human sperm cells. Reproductive functions in the male, it's controlled mostly by the hypothalamus, anterior pituitary, and the testes. And the functions of the hormones just to basically initiate, maintain sperm cell production, and oversee secondary sex characteristics. For the hypothalamus, we have gonadotropin releasing hormone, or GnRH. And GnRH regulates the secretion of your gonadotropins from the anterior pituitary. If you remember, the hormones of the anterior pituitary are kind of like intermediate hormones, are also called tropic hormones, where the hypothalamus secretes something that causes the anterior pituitary to secrete something which causes so the anterior pituitary secretes another hormone which acts on another gland to secrete something. So all these anterior pituitary hormones are kind of intermediate hormones that act on something else. So your anterior pituitary hormones, those are all called gonadotropins. Interstitial cell stimulating hormone, or ICSH, luteinizing hormone, LH. Those are going to cause your interstitial cells in your testes to secrete testosterone, or your male sex hormones. Your FSH, your follicle stimulating hormones. Typically, we associate FSH with females because it does say follicle stimulating hormones, but males do have FSH as well. FSH does stimulate the sustenticular cells of the testes to mature, proliferate, and respond to testosterone. Now, I said that LH secretes or helps secrete testosterone. And so if you have an increased plasma concentration of luteinizing hormone, that would stimulate the secretion or the release of testosterone. So in the presence of FSH and in the presence of testosterone, the sustenticular cells are going to stimulate spermatogenesis in the spermatogenic cells. And this is all a negative feedback loop. Remember, most of the body processes happen in a negative feedback loop. So once we have enough sperm production, or once we have enough sperm, we're going to, that sperm is going to say, hey, not obviously the sperm's not going to say, but the sperm is going to say, hey, we have enough sperm, so go ahead and drop off the concentrations of LH and FSH. So the hypothalamus is going to stop releasing GnRH, which is in turn going to stop the anterior pituitary from releasing ICSH, LH, and FSH. So these androgens, the testosterone, is going to prevent oversecretion of GnRH, LH, and I should say inhibits inhibition. That's inhibition from sustenticular cells prevents oversecretion of FSH. Actually, I lied again. We have this thing called inhibin. I just looked it up. Inhibin from it's actually a hormone from the sustenticular cells, prevents oversecretion. So I did have it right the first time. Inhibin prevents oversecretion of FSH. All right. Androgens are your male sex hormones. Remember, the interstitial cells of the testes are going to produce most of your androgens, but a small amount of these are made in the adrenal cortex. Remember, the adrenal cortex is what sits on the kidney, but testosterone is going to be your most important androgen. It's going to start in fetal development, continues during birth, but it's going to stop during childhood. But you're going to see, you're going to see testosterone kick back up during puberty, and going to continue through life. But it's testosterone is what's responsible for development, maintenance of all of your secondary sex characteristics. And we also have another androgen called dihydrotestosterone, or DHT. It's basically a derivative of testosterone. All right, so what does testosterone do? Basically descends your testes and develops the male reproductive organs. But during puberty, I said does your secondary sex characteristics, along with developing your accessory organs. So, I think what your or what the male secondary sex characteristics are. Increased body hair, sometimes you'll see decreased growth on scalp, deepening of your voice, thickening of your vocal cords, thickening of your skin, muscle growth, thickening strength of bones, increased rate of red blood cell production, and cellular metabolism. So that's basically pre-puberty, cellular metabolism. Now, I don't know if you've heard, but there's a condition called PCOS, polycystic ovarian syndrome. It's actually a disorder in females, where females produce abnormally high levels of androgens, particularly testosterone. It's actually going to result in females developing male secondary sex characteristics. So females can develop body hair, thick skin, muscle growth, strengthening of bones, deepened voice, stuff like that. All right, and this just shows, how we, the body and the negative feedback loop. There's our inhibin, our feedback loop of what we talked about how we go from GnRH to LH, FSH, stuff like that. So we're not going to rehash this slide. All right, so the reproductive organs in a female. We'll talk about a lot of these, your cervix, the vagina, vaginal orifice. There's the minor and major labium, the clitoris. Urethra is urinary. Now remember, in the male, your urethra is urinary and reproductive, but not in the female. Uterus, ovary, uterine tubes, ureter. All right, so for the female, the female sex cells are, remember, called eggs or oocytes. The sex organs of the female are going to transport the egg cells or the oocytes to the site of fertilization. The uterus is favorable. It has a favorable environment for development of offspring, and it transports that offspring to the outside of the body. That sounds very clinical. Can't we just say, pregnancy and delivery, but produces sex hormones as well. Female reproductive organs, we have our primary ones that are the ovaries. Those are the gonads. And we also have the secondary or accessory sex organs. The females have internal and external secondary reproductive organs as well. The ovaries, ovaries are solid structures. They lie in the wall of the pelvic organ. You can kind of see that they are pretty much kind of attached to the hip bones there. But they are also almost like a spider web thing, that they have a lot of ligaments that are gonna support them and keep them in place. There's really three big ligaments that kind of are involved with the ovary. The first one is the broad ligament. It's going to be the largest ligament. There's your broad ligament there. And it's attached to the uterine tubes and the uterus. And then you have the suspensory ligament. There's the suspensory ligament. And that's just going to hold the ovary in place on the superior end. And then you have the ovarian ligament right there that's going to hold the inferior end of the ovary to the uterus. All right, so the structure of an ovary, we can divide it usually into like two different sections. There's the medulla and the cortex. Remember, medulla kind of means middle layer, or in this case, an inner layer. Has a lot of loose connective tissue, areolar connective tissue, with blood vessels, lymphatic vessels, nerve fibers. And then the outer layer, the cortex, has a more compact tissue, more granular appearance. So the cortex itself would just be kind of like this outer layer, just kind of like the shell, and this medulla would be this whole kind of inside. All right, descent of the ovaries, just like in the male. The ovaries develop from the tissue from the parietal peritoneum near the kidneys, and then the ovaries descend where they're going to attach to the pelvic brim. All right. Now, primordial follicles. So during prenatal development, so before females are born, we have these things called primordial germ cells. They're also called oogonia. And they're going to divide by mitosis, not by meiosis, but by mitosis to produce more oogonia in the unborn fetal ovaries. And these oogonia develop into primary oocytes. All right. And then the primary oocytes, they're just surrounded by a bunch of epithelial cells called follicular cells, and they form this primordial follicle. And then in fetal development, these primary oocytes start meiosis, but then stops all of a sudden, and it doesn't really start again until puberty. And once the primordial follicles are produced in fetal development, no new ones are produced. So during fetal development, you're going to have all of the eggs in your body that you're going to have for the rest of your life. And then when you hit puberty, those eggs are going to start to kind of develop and I can't think of the word, are going to develop and become nourished and continue on with the rest of with meiosis. So you're going to produce about 300 to 400 eggs during or oocytes during a female's lifetime, and all of those are produced or made during fetal development. But at some point in fetal development, that the production of oocytes kind of stops, and or the development of those eggs stops, and then it'll pick up again in puberty. And then it says number of oocytes continually declines, and 90% of the primordial follicles present at birth are lost to apoptosis between birth and early childhood. So apoptosis, remember, is like natural programmed cell death. So like if a red blood cell needs to go die off, it's going to die off through apoptosis. So same thing with oocytes. I guess I should back up and say 300 to 400 oocytes are released, not produced. So let's just do the math. So let's just say, if we use 400, so you're actually 400. So if females release 400 eggs, and it says 90% of them will be lost due to apoptosis, during fetal development, you actually produce about 4,000 eggs. And then if you lose 90% of them, 4,000 minus 90% is going to be 400 eggs. All right, so some of your internal accessory organs, uterine tubes, uterus, and vagina. Uterine tubes are also called the fallopian tubes, or sometimes you'll hear them called the oviducts. They're just a tubular organ that transports the egg from the ovary to the uterus. And I said earlier that fertilization normally occurs in the fallopian tube or the uterine tube. It does have three layers: the inner mucosa, a middle muscular layer, and then outer covering is a peritoneal layer. Has a bunch of cilia in them. Remember, cilia is going to be for transport of egg down the uterine tube into the uterus. But one key function is that it also has a peristaltic movement so that it will help the movement of the egg in addition with the cilia. All right, this is just a diagram of female reproductive organs. You can see the body of the uterus with the endometrium, the myometrium, and the parametrium. Your cervix actually, with your vagina, your cervix, you can see here, your cervix, actually about a third of your cervix hangs down into your vagina. There's your uterine tube or your fallopian tube. There's your ovaries. All right. I thought these colors were pretty. Anyway, so these are just some cilia. These are just basically your uterine tubes. You can see the cilia that are present that will help move the egg down the uterine tube. All right, the uterus itself, it's a hollow organ, and that it receives the embryo, sustains development. Once fertilized, the embryo should attach to the uterus, but the uterus itself is attached to the pelvic wall by the broad ligament and the round ligament. Remember that the broad ligament also holds the ovary in place. We can see that there's different parts of the uterus. There's the body of the uterus, the cervix as well. The body of the uterus is the upper 2/3 of the uterus. So the body is that upper 2/3. And the neck of the cervix is that lower 1/3. The body does contain, it's called that fundus. So up here is that fundus area. For those of you going to nursing school, when you do a vaginal birth, you actually have to go in and massage the fundus, and that will actually help expel any of afterbirth that might still be in the uterus, like if you have any, retained blood clots or anything like that. You have to massage that fundus to kind of help get rid of all that gunk that still might be in the uterus. The cervix itself, it's the lower third, partially extends into the top part of the vagina. That's what practitioners will use for when they do a pap smear to look for, any STDs or cervical cancer, stuff like that. We already talked about the layers of the uterine wall, the endometrium, the myometrium, the paramecium. Here you can see the uterine wall. All right, so the vagina itself, it's a very muscular tube that runs between the uterus and the outside of the body. So here, we can't really see it, but you can see that those rugae or those folds in the vagina. So it's very muscular. It conveys uterine secretion, receives the penis during intercourse, provides a passageway for offspring during birth. It surrounds the ends of the end of the cervix. Remember the cervix, about a third of the uterus. falls into the vagina. There's a vaginal orifice that's partially enclosed by a hymen. Remember, I don't know if you know, the hymen is a thin layer of connective tissue and stratified squamous epithelium. And usually during the first time you have intercourse, that hymen is broken and can cause some unpleasant feeling and some bleeding. Just like the uterus, the vagina has 3 layers: an inner mucosal layer, middle muscular layer, and outer fibrous layer. Some of your female external accessory reproductive organs. The vagina and are also called the vulva. Surround openings of the urethra. The urethra is anterior to the vagina and are called the vulva. So collectively, it's the vulva. Some of the organs that are include the labia majora, or the labia major, the labia minor, the labia minora, the clitoris, and the vestibule. So the labia majora are rounded folds of the skin that are more near the symphysis pubis or the mons pubis. The labia minora are more at the anterior end, and they form like a hood-like covering over the clitoris. The clitoris is between the labia minora at the anterior portion of the vulva. Clitoris is analogous to the male penis. It's a sexual arousal portion of the female. Has 2 columns of erectile tissue. And then there's a vestibule, which is a space between the labia minora, and it encloses the vaginal and urethral openings. So here you can see the labium majora. So that's just kind of like on the outside. The labia minora would be on more like the inside. There's the vestibule gland is also I guess the labia minora is, I labeled it wrong. The vestibule gland is more on the inside. And then your clitoris is here. That's the erectile tissue. That's the analogous or very similar to the male penis. Now for females, females do go through a similar process, erection, like males do. Like I said, their erectile tissue is the clitoris, or clitoris. There's multiple ways to pronounce it. Clitoris. And it's in response to sexual stimulation. Just like in males, nitric oxide is going to dilate those arteries. And I believe I said this before in previous lectures. Viagra works by nitric oxide release. So that's why people take Viagra, is because it stimulates nitric oxide release. All right, so lubrication kind of works the same way. There's sexual stimulation causes those vestibular glands to secrete mucus into the vestibule, which is going to lubricate the vagina to aid in intercourse. Just like males, females can experience orgasm. The clitoris does respond to sexual stimulation, ends with orgasm, which again is a pleasurable feeling, a physiological, psychological release. And the muscles of the perineum, uterus, and uterine tubes all contract rhythmically. And there is a biological purpose to this. The goal of biology, or the goal of life, uh, is for reproduction. And when females orgasm, what the body says is, oh, I need to collect all that sperm and bring that sperm up to, as close to the fallopian tube where the egg is going to be, as close as possible. So all of these muscle contractions are going to help transport that sperm towards the fallopian tubes, so that the sperm doesn't have to do as much work. All right. Again, just a summary slide of what we just talked about and some functions of the reproductive organs. All right, oogenesis and the female reproductive cycle. So remember I said that, that we talked about how, there's meiosis 1, meiosis 2, and sperm, goes through the meiosis 1, meiosis 2. But with eggs, remember how I said that with oocytes, in fetal development, eggs are produced, but then meiosis 1 all of a sudden stops. And then sometime during puberty, meiosis 1 will pick up again. So beginning at puberty, some of those primary oocytes, because remember primary, we're still in meiosis 1, we're going to still have we're going to continue meiosis 1. At the end of meiosis 1, we have telekinesis, and telekinesis is where, the cell divides. So we're going to have 23 chromosomes. So we're going to have half as many chromosomes as the parent cell. However, that cell doesn't split right down the middle. A cell doesn't split 50-50. That cell will split about 90% will go on one side and 10% will go on the other side. So that cytoplasm that's in the cell, 90% of that cytoplasm will go on one side and 10% will go on the other. So let's say that this is your parent cell. When we have cytokinesis, let's say that cell is going to split like this, so that we're going to have two different cells. One cell that's this big and one cell that is this big. That's different from spermatogenesis, where the cytoplasm is going to divide equally and we're going to have two cells that essentially looks the same. In eggs or in oocytes, this is different. The cells are going to look very different. But you're still going to have the same genetic information in this cell as you do in this cell. It's just the amount of cytoplasm is going to be different. This new cell is called a polar body. I'm just going to abbreviate it, polar body. All right, so we have that, and we have the secondary oocyte and a polar body. Now, sperm will fertilize, most likely, this secondary oocyte. polar bodies really are there for are there to provide some organelles for the secondary oocyte, some just some backup stuff. But in some cases, a sperm can fertilize that polar body. Now, it might not develop into a full fetus because there's not as much cytoplasm, there's not as much nutrients as this secondary oocyte. So it might result in a miscarriage, or it might not, the cell might not even implant in the uterine wall. But, the key takeaway is that, the polar body does have the exact same genetic information as the secondary oocyte does. But it doesn't have the large amount of cytoplasm, the large amount of nutrients, and the large amount of organelles that the secondary oocyte has. All right, so this is just a picture of oogenesis. We're not going into this in depth. This definitely goes way more in depth than we need to go into. So here's just like a picture of the secondary oocyte and the polar body. So you can see how the size difference of the polar body is compared with that secondary oocyte. All right, follicle maturation. So once females hit puberty, that anterior pituitary gland is going to release a ton of FSH, follicle stimulating hormone. And that follicle stimulating hormone is going to go down to the ovaries and is going to stimulate the ovaries to do their thing. And if you haven't hit menopause yet, during reproductive years, those primordial follicles are going to now develop into primary follicles. Remember, there's 300 to 400, and you're not going to mature all 300 to 400 eggs at once. You're going to mature 1, 2, 3 eggs at a time. And that primary oocyte is going to enlarge. Those follicular cells are going to proliferate. And then we're going to see this glycoprotein layer. It's called a zona pellucida layer form between the oocyte and the granulosa cells. And as that follicle is developing, ovarian cells are going to start to become organized into different layers. We're going to be able to see this inner vascular layer and outer vascular layer. It's called a theca interna and a theca externa. This inner layer is going to produce steroids. That outer layer is going to produce a connective tissue. And it's going to eventually proliferate into up to in between 6 and 12 layers long. And between the cells, it's going to have a bunch of fluid in between those cells. And it's going to take about 150 days to develop those primordial follicles to mature, develop. And once they do, we're now going to call them preantral follicles. So here you can just see the maturation of the primordial follicle into the preantral follicle, and then the mature, and then eventually females will ovulate. All right, so here just showing the different stages of follicular maturation. Here's your oocyte. Here's a maturing antral follicle, a preantral follicle. All right. So for follicle maturation, as the follicle maturates, that oocyte's going to become pressed to one side of the follicle. So it takes 150 days to go from the primordial eraser. So it's going to take 150 days to go from the primordial follicle to the preantral follicle. And then it's going to take another 65 days to go from a preantral follicle into a mature antral follicle, and then 15 more days to go from an antral follicle into a mature antral follicle. So 70 plus 15 would be about 85 more days. And then, oh, no, I said one to 3 eggs. 20 eggs. So 20 primordial follicles can begin maturing at one time, but only one dominant follicle usually is going to outgrow the others. So even though females are going to start out with several eggs that are maturing, typically only, they're going to start out with, 20 eggs that are maturing, only 1, 2, 3 eggs are going to get released. And only the dominant follicle that fully develops, while others are going to degenerate and get reabsorbed back into the body, because the body doesn't like to discard anything that it can reuse. So this is a lot of protein that the body can reabsorb. And then it takes about 300 days for the primordial follicle to develop into a mature antral follicle. And then once the mature antral follicle is ready for ovulation, that's when we have the menstrual cycle. I guess I didn't develop or advance your slides. Sorry about that. All right. Here you can see the mature antral follicle. All right. Ovulation. So once the follicle matures, that primary oocyte undergoes meiosis 1, producing that secondary oocyte and that polar body. So we're going to produce that primary oocyte and that polar body. And then that ovulation releases that secondary oocyte and that first polar body. And it's going to happen, ovulation happens because of that LH that's going to be released from the anterior pituitary gland. Once the anterior pituitary gland releases LH is going to act on the ovaries so that the follicle swells, weakens, ruptures. That secondary oocyte and those surrounding follicular cells are propelled to the opening of the uterine tube by these things called fimbriae, and then once that oocyte is released, it's going to need to be fertilized within hours, or it's going to degenerate. Now, I've had people ask questions. Well, how do I know? When I actually ovulate. Some people know and some people don't know. But, sperm can live in the female body for up to 3 to 5 days. So if you've had sex, let's say because you're on birth control. Let's say you know you're going to ovulate on Tuesday, and you had sex on Sunday, and if there's still sperm inside of your body, that egg might be fertilized. Yeah. So, on average, sperm will live in your body for up to 5 days, with the average being about 3 days. All right. Here's just a picture of your secondary oocyte as it's getting expelled from the ovary. All right, so female sex hormones, or your estrogens, remember they're going to be released by the hypothalamus, go through the anterior pituitary gland, and so the estrogens, just like with the male, the estrogens are gonna allow for female secondary sexual characteristics, maturation of female sex cells, but also changes during the menstrual cycle. We have GnRH, just like we do in the males, secreted by the hypothalamus, which causes the anterior pituitary glands to release FSH and LH. All right, in females, FSH and LH, they're going to control oocyte maturation and production of the female sex hormones. Remember, I talked about PCOS, that polycystic ovarian syndrome, and hormone changes. You're actually going to see hyper secretion of LH, so too much LH, as well as hypo, or too little, hyposecretion of FSH. Hypersecretion of LH is going to cause abnormal production of androgens in females. All right, estrogens and progesterone. They're secreted by the ovaries, adrenal cortex or cortices, and the placenta during pregnancy. Estrogens are going to stimulate the accessory reproductive organs, uterine wall, during development. And they're going to develop your female secondary sex characteristics. All right, so someone knocked on my door, and I have no idea why it was. So estrogens, I think we're on secondary sex characteristics, breast development, mammary gland development, adipose tissue in the breast, thighs, buttocks. Progesterone stimulates uterine changes during menstrual cycle, regulation of your gonadotropins. Also, females have the androgens like testosterone, the dihydrotestosterone, DHEAS and DHEA is secreted by the adrenal cortex, which causes axillary hair to grow in the pubic region, or hair to grow in the axillary region and the pubic region. Kind of like the similar slide that we saw earlier. All right, so your menstrual cycle. On a regular, reoccurring basis, it's marked by changes in the endometrium of the uterus and culminates with menstrual bleeding. We call that menses. It usually begins around age 13 and continues up until around 50 or early 50s. Your first period, or your first menstrual cycle, is called menarche. And like I said, it's usually each month at a regular interval. As one antral follicle matures, it's ovulated, and the remnants of that follicle closes up to become the corpus luteum. Now, in the absence of fertilization, that egg cell or that oocyte is going to degenerate, and that corpus luteum is going to degenerate as well to become this thing called a corpus albicans. All right, so there's different phases of the menstrual cycle. There's the in the, I guess, two big phases. There's an ovarian phase and a uterine phase. But in the ovarian phase, we can break it down into the follicular phase, ovulation, and the luteal phase. In the follicular phase, this is kind of directed by FSH. Ovulation is triggered by LH. Remember, LH comes from the anterior pituitary gland, which, remember, LH is stimulated by GnRH from the hypothalamus. And so ovulation is the final development of the mature antral follicle, and ovulation is the release of that oocyte or egg cell from that surrounding layer from the follicle and ovary. That luteal phase is that corpus luteum that forms from the remnants of the follicle, and it secretes the estrogen, progesterone, and inhibin. If the egg cell is not fertilized, then that oocyte will degenerate in about 12 days and become a corpus albicans. Now, in your uterine phase, there's three phases: the proliferative phase, the secretory phase, and the menstrual phase. Now, these aren't like two separate phases. They're the same. They go hand in hand, but you can look at it kind of like what happens physiologically and more what happens anatomically. Those aren't the right words, but you can see like what happens from a hormone perspective or what happens from a, muscular perspective. In the uterine phase and the proliferative phase, you can see that the endometrium starts to thicken up as a result of increased estrogen secretion. In the secretory phase, that endometrium becomes more vascular because of progesterone, and the uterine glands are going to produce a lot of nutrients in preparation of that egg possibly being fertilized. And then the menstrual phase, that endometrial lining is going to slough off once the corpus luteum degenerates, and that menstrual bleeding is going to remove that sloughed off tissue. All right, so here you can see the hormone control of female secondary sex characteristics. We already talked about GnRH that stimulates the anterior pituitary gland to act on FSH and LH. So we kind of already covered FSH, LH, and the estrogens. Here, the events in a menstrual cycle. I'm going to do kind of reformat it just a little bit. There we go. Make it a little bit easier to read. All right. Okay, and also here you can see a picture version of what happens in the menstrual cycle. Female infertility, it's usually defined as the inability to conceive a child after 1 year of trying. It's not a female fault. It's not a male fault. Both females and males contribute to infertility equally. 25% of the cases, you can't isolate one factor as to why a person or a female is infertile. Some of the causes could be anovulation, could be hyposecretion of LH and or FSH that results in lack of ovulation. It could be endometriosis. And endometriosis is just growth tissue that builds up in the abdominal cavity that affects the uterine lining, could become fibrotic to prevent ovulation, and it obstructs the uterine tubes. It could be the result of an infection. Maybe the female had gonorrhea earlier on in life. That can cause an obstructed uterine tube. Now, another thing that can contribute to infertility is not necessarily a permanent effect, but exercise. If you look at, gymnastics, a lot of younger gymnasts don't get their period until later on in life once they quit gymnastics because they are exercising frequently. Lots of exercise can contribute to infertility. The hardcore exercise can lead to decreased secretion of GnRH. And if you have decreased secretion of GnRH, that's going to lead to decreased secretion of FSH and decreased secretion of LH, which would lead to decreased ovulation. So that hardcore exercise or lots of exercise could lead to infertility issues. All right, menopause. It's also called female climacteric. It's usually the stoppage or the stopping of menstrual cycles for 1 year. Usually you menstruate until you're in your early 50s, late 40s, and you stop ovulating because of the age of your ovaries. And it's because there is an increased gonadotropin concentration as well as a decreased estrogen concentration in the bloodstream. And you'll see signs of some of your female secondary sex characteristics start to disappear. You might see shrinkage of the vagina, the uterus, and possibly your breasts, breast tissue. With the hormonal changes that come with menopause, you could see osteoporosis. Which is, increased loss of bone matrix, which is why a lot of people on menopause have to take calcium and vitamin D supplements. Because of the hormonal changes, a lot of women, more than 50% of women, experience hot flashes. Migraines, headaches, backaches, fatigue are some more signs and symptoms, and hormone replacement therapy can help minimize some of these symptoms that you feel. All right, mammary glands. They are accessory organs, and they are specialized organs that secrete milk following pregnancy. Obviously, the purpose is to nourish baby. They are located within the breasts in the anterior thorax, and they have a nipple that's surrounded by circular area called the areola. And each mammary gland has 15 to 20 irregularly shaped lobes. And that each lobe has this alveolar gland. Here you can see the alveolar glands. You can see the lobes. So each lobe has this alveolar gland, which are drained by the alveolar ducts, which drain into this lactiferous duct that is going to drain to the outside. And that's what how baby gets milk. And the alveolar glands produce milk in a lactating female. And these dense strands of connective tissue are in the mammary glands that support the breast. Now, males and females have very similar mammary glands, but it's not until puberty that we start to see the differences in the structure. That ovarian hormones stimulate the development of the mammary breasts in female. But structurally, both males and females have very similar breast structure. With breast cancer, some of the warning signs: thickened tissue, dimples, change in color, contour or change in contour, a nipple that flattens or points in abnormal direction, nipple discharge. If you do suspect something, please go see your doctor, nurse practitioner, PA, whatever. You can get a lumpectomy after, they're going to do a biopsy, but they might do a lumpectomy, a simple mastectomy, a radical mastectomy, chemotherapy, radiation. But some of the newer drugs, some estrogens, stuff like that, that can block. Newer drug signals from stimulating cancer cells to divide. So basically what we're doing is we're blocking estrogen receptors. And 5 to 10% of breast cancers arise from inherited tendency. So genetic information or genetic testing can be beneficial to women who have possibly inherited certain genetic variants. But one thing that's interesting to note is that about 1% of all breast cancer cases are men. All right, birth control. Birth control is a voluntary regulation of the number of offspring. It requires some type of contraception, which basically avoids fertilization following sexual intercourse or preventing implantation. And we have different methods of birth control with varying degrees of effectiveness. So there's coitus interruptus, which basically this is for a lack of a more common term, is the pull-out method. It's the practice of withdrawing the penis before ejaculation. And the theory behind it is it prevents the entry of sperm into the female reproductive tract. However, it's not very effective because sperm do leave the urethra before ejaculation. There's sperm in the pre-ejaculatory fluid, so pulling out before ejaculation is not an effective method because there's already sperm in your pre-ejaculatory fluid before the man ejaculates. The rhythm method. And it says here, requires abstinence from sexual intercourse 2 days before and 1 day after ovulation. Well, this isn't very effective because one, you don't know the exact time of ovulation. And two, 2 days before and 1 day after ovulation, if one, you don't know the exact time of ovulation, and two, sperm can live inside the body for 5 days. So, if you're only going 2 days before and 1 day after ovulation, let's say you do know the exact day and the exact time, you're only covering, if you ovulate on the 10th and you had sex on the 5th, that sperm is still viable. Or if you ovulate on the 5th and you have sex on the 15th, you can still get pregnant. So you'd actually have to do the rhythm method, for 10 days if you know the exact date that you're going to ovulate. So again, not very effective. Mechanical barriers, these are probably the most often used. Male condoms are probably the most often used, but there are female condoms as well, and they just prevent sperm from entering the female tract during sexual intercourse. Chemical barriers, spermicides, creams, foams, jellies. These have actually gone down in popularity just because most people don't even know about these. I actually talked to my class last semester about this, and they had no idea what I was talking about. But just know that there are other forms of birth control out there just besides, condoms. But they are most effectively used, or they are most effective when used with condoms as well. Birth control pills is the most common combined hormone or oral contraceptive. It delivers estrogen and progesterone to prevent pregnancy, and there's synthetic chemicals that function like estrogen and progesterone, and they essentially disrupt the normal FSH and LH secretion that prevents follicle maturation and ovulation. They also cause the cervical mucus to prevent sperm passage. There's some other hormone contraception out there. You can get an intramuscular injection that's good for 3 months. There's IUDs or intrauterine devices that a practitioner can put inside your uterus. Let's see, there's emergency contraception that is typically called the morning after pill. It usually can be taken 3 to 5 days after intercourse. It works by stopping ovulation. It prevents pregnancy, but if ovulation has already occurred, obviously it will not prevent that ovulation because ovulation has already happened. And then now it's more common to see sterilization procedures. And it says surgical procedures that permanently prevent pregnancy. And I'm actually going to put permanently in quotation marks because permanently is not permanently. They can always be reversed. Vasectomy in males is basically where you snip the ductus deferens, or that vas deferens, and it prevents sperm from leaving the epididymis to go into the urethra, but it does not affect any of the interstitial cells. And there's also a tubal ligation for the female where the uterine tubes, or those fallopian tubes, are cut and the ends are tied. And that prevents sperm from reaching the oocyte. So here you can see that on the male side, basically we're snipping right here, the vas deferens, and in the females, we're going to snip right there. All right, so here you can see the various methods of birth control. What I found interesting is that if you use condoms, they are only 88 to 98% effective. And I asked my class last semester why they thought that condoms were only up to 80, or they were 88 to 98% effective. And they came up with a lot of varying answers. One of the answers was, oh, well, we had sex without a condom, but we only used a condom when the guy ejaculated. And I'm like, well, that's stupid. Why have the condom on at all? Because there's sperm in the pre-ejaculatory fluid. Anyway, just, and of course you don't have to know any of the numbers or anything, but you do have to know, just the different methods. So here, the pull-out method or the withdrawal method, it's only, 82% effective. All right, so sexually transmitted infections. So we need to say sexually transmitted infections instead of saying sexually transmitted disease. So we don't say STD anymore, we say STI. Now, because a person can be infected and contagious without developing symptoms of a disease. So some of the symptoms of a STI, most of these you should know already. You know, a burning sensation during urination, maybe pain in the lower abdomen, a fever, discharge from the discharge from the penis or vagina, painful itching or inflammation, sores, bumps, blisters, rashes on the genitals, itchy or runny eyes. I thought that was an interesting one, but you can actually have a sexually transmitted infection in your eye. So, all right, pelvic inflammatory disease. It's usually associated with gonorrhea or chlamydia. It's basically where bacteria enters the vagina, spreads to other reproductive organs, and it can scar your uterine tubes and lead to infertility. Acquired immune deficiency syndrome, AIDS, that's the end result of HIV if left untreated. HIV is transmitted through semen, unprotected sex, blood, or mother's milk. And basically it just deteriorates the body's immune system. With HIV, you will never have to know, the number of cases reported. So, you're not going to have to know this column here, but you will have to know the signs and symptoms of HIV. You will have to know everything about genital warts, with the exception of number of cases reported. Gonorrhea, syphilis, herpes. Again, you will not have to know the number of cases reported, but you will need to know everything about gonorrhea, syphilis, or everything to know about gonorrhea. All right, so that brings us to the end of chapter 22, the end of AMP2. So if you have any questions, please let me know. Otherwise, take care, and it's been fun. Bye.